வணக்கம். Welcome to NCERT Biology Reading, Class 11, Chapter 9, Biomolecules, Introductory Part. There is a wide diversity in living organisms in our biosphere. Now, a question that arises in our mind is: are all living organisms made of the same chemicals, that is, elements and compounds? You have learnt in chemistry how elemental analysis is performed. If we perform such an analysis on a plant tissue, animal tissue, or a microbial paste, we obtain a list of elements like carbon, hydrogen, oxygen, and several others, and their respective content per unit mass of living tissue. If the same analysis is performed on a piece of Earth's crust as an example of non living matter, we obtain a similar list. What are the differences between the two lists? In absolute terms, no such differences could be made out. All the elements present in a sample of Earth's crust also present in a sample of living tissue. However, a closer examination reveals that the relative abundance of carbon and hydrogen with respect to other elements is higher in a living organism than in Earth's crust. Table 9.1. Then 9.1, how to analyze a chemical composition. We can continue asking in the same way, what type of organic compounds are found in living organisms? How does one go about finding the answer? To get an answer, one has to perform a chemical analysis. We can take any living tissue, a vegetable or a piece of liver, etc., and grind it in a trichloroacetic acid, Cl3CCOOH. Using a mortar and pestle, we obtain a thick slurry. If we were to strain this through a cheesecloth or cotton, we would obtain two fractions. One is called the filtrate, or more technically, the acid soluble pool, and the second, the retentate, or the acid insoluble fraction. Scientists have found thousands of organic compounds in the acid soluble pool. In higher classes, you will learn about how to analyze a living tissue sample and identify a particular organic compound. It will suffice to say here that one extracts the compounds, then subjects the extract to various separation techniques till one has separated a compound from all the other compounds. In other words, one isolates and purifies a compound. Analytical techniques, when applied to the compound, give us an idea of the molecular formula and the probable structure of the compound. All the carbon compounds that we get from living tissues can be called biomolecules. However, living organisms have also got inorganic elements and compounds in them. How do we know this? A slightly different but destructive experiment has to be done. One weighs a small amount of living tissue, say a leaf or liver, and this is called wet weight and dry weight. Wet weight and dry it. All the water evaporates, the remaining materials give dry weight. Now, if the tissue is fully burnt, all the carbon compounds are oxidized to gaseous form, that is carbon dioxide and water vapor, and are removed. What is remaining is called ash. This ash contains inorganic elements like calcium, magnesium, etc. Inorganic compounds like sulfate, phosphate, etc. are also seen in the acid soluble fraction. Therefore, elemental analysis gives elemental composition of living tissues in the form of hydrogen, oxygen, chlorine, carbon dioxide, carbon, etc. While analysis for compounds gives an idea of the kind of organic and inorganic constituents, table 9.2, present in living tissues. From a chemistry point of view, one can identify functional groups like aldehydes, ketones, aromatic compounds, etc. But from a biological point of view, we shall classify them into amino acids, nucleic acids, nucleotide bases, fatty acids, etc. Amino acids are organic compounds containing an amino group and an acidic group as substituents on the same carbon. That is, the alpha carbon, hence they are called alpha amino acids. They are substituted methanes. Hence they are called alpha amino acids. They are substituted methanes. There are four substituent groups occupying the 4 valency positions. These are hydrogen, carboxyl group, amino group, and a variable group designed as R group. Based on the nature of R group, there are many amino acids. However, those which occur in proteins are only of 20 types. The R groups in these proteinaceous amino acids could be hydrogen. The amino acid is called glycine. A methyl group alanine, hydroxymethyl group which gives serine, etc. They 3 of the 20 are shown in figure 9.1. The chemical and physical properties of amino acids are essentially of the amino, carboxyl and or functional group based on the number of the amino acid and carboxyl group. There are acidic, example glutamic acid, basic glycine, and neutral valine amino acids. Similarly, there are aromatic amino acids, tyrosine, phenylalanine, tryptophan. A particular property of amino acids is ionizable nature of NH2 and COOH groups. Hence, in solutions of different pH, the structure of amino acid changes. Lipids are generally water insoluble. They could be simply fatty acids. A fatty acid has carboxyl group attached to the R group. The R group could be a methyl CH3 or ethyl CH2 C2H5 or higher number of CH2 groups, 1 carbon to 19 carbons. For example, palmitic acid has 16 carbon including carboxyl carbon. Arachidonic acid has 20 carbon atoms including the carboxyl carbon. Fatty acids could be saturated without double bond or unsaturated with one or more C double bond C. Another simple lipids is glycerol, which is trihydroxypropane. Many lipids have both glycerol and fatty acids. Here, the fatty acids are found esterified with glycerol. They can be then monoglycerides, diglycerides, and triglycerides. These are also called fats and oils based on melting points. Oils have lower melting point, example gingerly oil, and hence remain as oil in winters. Can you identify a fat from the market? Some lipids have phosphorus and phosphorylated organic compounds in them. These are phospholipids. They are found in cell membrane. Lecithin is one example. Some tissues, especially the neural tissues, have lipids with more complex structures. Living organisms have a number of carbon compounds in which heterocyclic ring can be found. Some of these are nitrogen bases: adenine, guanine, cytosine, uracil, and thymine. When found attached to a sugar, they are called nucleosides. If a phosphate group is also found esterified to the sugar, they are called nucleotides. Adenosine, guanosine, thymidine, uridine, and cytidine and nucleosides. Adenyllic acid, thymidyllic acid, guanidinic acid, uridylic acid, and cytidyllic acid are nucleotides. Nucleic acids like DNA and RNA consist of nucleotides only. DNA and RNA functions as genetic materials. Structures of some biomolecules: sugars, amino acids, then fats and oils, nitrogen bases: adenine, adenosine, adenylic acid, uracil, uridine, and uridine. Thank you.